Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this session, we're going to talk about chromosomes and cell division, and we're going to have a particular emphasis on the cell division process called mitosis. Chromosomes, let's give a little bit of historical background and sort of catch up to where we were. Remember, Mendel was very good at doing crosses and collecting data and describing how traits were passed on from parents to offspring. However, he really had no idea what his genes or factors were made of, where they were located, and nor could he. It's okay, I'm not criticizing him. However, it turns out that chromosomes are the carriers of the genetic information, and we're now going to try and look at how chromosomes behave, what they are, and try and integrate that material with what we already know from Mendel's work. So here's a rather beautiful, I think you'll agree, picture of some chromosomes. These are chromosomes, all of them sort of look like an X, and we'll see why in a moment. They don't all really look quite as symmetrical as this. This is a bit of a simulation. But when we envision them and when we see them under the microscope, they do take on this type of an appearance that looks like an X. They really are quite beautiful. So, just to recap, Mendel's laws certainly seem to be true. They seem to be consistent, and all was well with the world in terms of the Mendelian world. But as I just said, sometimes we want to pretend for the sake of argument that we don't know things. But at this point, almost everybody, even non-scientists, know that genes are in fact made up of DNA. He had no idea about that. He also did not know they were located on the chromosomes in the cell. The major thing that his work did allow was a prediction of the outcomes of crosses, assuming that you knew, dominance relationships and things of that nature. So Mendel's work was rediscovered around 1900, and just about that same time, a discovery usually attributed to a scientist named Sutton was that the genes of Mendel were in fact located on chromosomes. Now, chromosomes are structures that are visible under even a reasonably powerful microscope, and they're located in the nuclei of eukaryotic cells. So peas and humans are both eukaryotes, at least we have that in common. Now, guys, it was known that chromosomes existed prior to this just because people, cytologists who looked under microscopes, saw them. But the connection was never made that these chromosomes and the genes really were very, very intimately connected. The way he did it, Sutton, and there were other people involved, basically is he looked at the way chromosomes were moving during cell division. And this is something we're going to talk about today. And what he realized is that they behaved exactly in the same way that Mendel's laws would predict if Mendel's factors, what we call genes, were in fact located on these chromosomes. So it's almost looked like the chromosomes were the factors in a sense. We'll see why, but basically this was the first time you could say that genetic analysis and cytology is just the study of cells, looking at cells, came together so we could make this really important connection about not just how the information behaved, but where it was and what it was to a point. And I also just wanted to mention that, now we know, in essence, it's a little oversimplified, but chromosomes are really just long molecules of DNA that are condensed to various degrees. So it's going to take us a while to get to the molecular part, the DNA part, but for now, let's think about chromosomes. And we're not going to continue to emphasize it here, but keep in the back of your mind that this is goes along with what Mendel was describing, even though it's not strictly about the information itself. The chromosomes, now we're looking more at their structures and how they behave. Okay, so we are going to focus on cell division today, and one of the reasons is because Think about this. We talked about in the pea plant experiments and even in our human pedigrees, information was being transmitted from a parent organism to its offspring. Now, that's reproduction, but cell division is a form of reproduction, and I dare say all forms of reproduction involve cell division one way or the other. Think about it this way, and maybe it'll be clearer. If you were a single-celled organism, well, you probably wouldn't be watching this video right now, but if you were a single-celled organism like a protozoan, the cell is the organism, right? And if that cell divides, we'll talk about the mechanism, you now got two new organisms. That is reproduction, right? And traits are being passed from that parent. It's not exactly the same, obviously, as the way we do it, from the original cell into the so-called offspring cells. So Mendel could maybe predict the way the traits would go and the ratios. Now we want to see how cell division actually works, which is sort of a reinforcement of how the information is flowing, given that the information is located on these structures. So some terminology that we need to be aware of. Adult eukaryotic organisms, whether that be you or I, whether that be an adult pea plant, we have a diploid chromosome number. You may be familiar with the term diploid. Algebraically, diploid is 2n. We'll discuss that briefly in a moment. But that's the chromosome number. So in humans, 2n happens to equal 46 in humans. That really is not a magic number. Humans happen to have 46 chromosomes. Pea plants, it turns out, have 50. There's really no real rhyme or reason to the exact number that any particular organism has. But it also means you can look at our complement of 46 chromosomes in two different ways, I think. One of those ways is to say that we have two complete sets. And those sets, each one is called a haploid set. It doesn't really come from this, but I think of sometimes haploid meaning half of the total. It's not just any half, but it's one complete set. In other words, if we use humans as an example, through your father's sperm, you received one complete set, and through your mother's egg, you received another complete set. So one complete set is algebraically known simply as n, and that gives you the idea that our diploid number, 46, is the combination of these two haploid complete sets. So I hope perhaps it's obvious that if 2n equals 46 in humans, n would equal 23. Okay? So that's one way you can think of it, is that our 46 chromosomes are really two sets of 23, two complete sets. However, I mean, it doesn't change what they are, but there's another way you can think of it, and that is that we have 23 individual pairs of chromosomes. Okay, because what happens is within the set, we number the chromosomes arbitrarily based on their size, 1, 2, 3, 4, 5, 1 being the biggest, 22 being the smallest. So another way of looking at this is that you have two chromosome number ones, perhaps obviously since they're part of the set, but you got one number one from your father, one number one from your mother. And the two number ones are similar in a variety of ways. So they're referred to as homologous chromosomes or homologs or even homologous pairs because they are a pair. So I guess my bottom line is you can think of it as having two sets of 23 or 23 pairs of homologs. It is what it is, as they say, but those are two different ways to envision it. So those two members, we're going to see that the homologs, sometimes it matters that they're homologs, sometimes it's not particularly that important. Now, in sexually reproducing organisms, which are almost all animals and plants, in fact, many animals and plants can reproduce either asexually or sexually, but Mendel's work focused on sexual reproduction. So let's use again humans as an example. There are two fundamental types of cell division, those being mitosis and meiosis. Some think it's a little bit unfortunate that the words sound so similar to one another, but that's sort of what we're stuck with. So before we get into the mechanics of what happens, let's talk about the rationale behind and the difference between mitosis and meiosis. Now, mitosis, again, using the example of an adult eukaryotic organism, mitosis occurs in so-called somatic cells. Somatic means body, but what it really means here is all the cells in your body that have nothing to do with sexual processes whatsoever, which is most of them, right? Nervous tissue, muscle tissue, etc., etc. So mitosis occurs there, and this is extremely important. The result of mitosis is two genetically identical so-called daughter cells coming from one so-called parent cell. Okay? So one way you could look at that is the parent cell is 2N, right? And after mitosis is done, you get two cells that are also both 2N. They are genetically identical to each other, and by the way, both of them are genetically identical to the parent cell as well. So in the very real technical sense, this is cloning. It's asexual. You can think of it as, see, I know you know this, but I have a daughter, so I reproduced and a new human organism came to be. But from cells in our body's point of view, think of this. When you grew from a baby up to an adult, we thought of one organism growing. But really what was happening was cells were dividing and more and more and more and more cells came into being. In the strictest, most technical sense, that is a form of asexual reproduction. All those cells are identical to each other and identical to their progenitors. Okay, so we think of mitosis in terms of various processes that occur in human beings, but it is fundamentally an asexual process. Whether you consider that reproduction or not, it's certainly, reproduction of cells. Meiosis, on the other hand, only occurs in cells that, the opposite. They're cells that are part of the sexual life cycle, and they're cells that are going to produce sexual cells called gametes. Gametes are sperm and eggs in, people and most other animals. Sometimes various other more exotic organisms, their gametes are called different things, but we can keep with that. So meiosis is intrinsically linked with a sexual life cycle. I don't mean the act of sex. I mean the point of a sexual life cycle. We'll talk more about when we talk about meiosis, but it's basically to make cells whose mission in life is to find another appropriate cell to meet up with and create a new recombinant organism. It results, meiosis that is, usually in 4 haploid gametes, being produced from one diploid parent cell. So it's very important to realize, with respect to this little diagram I drew here, you're basically starting with the same type of cell genetically, but instead of making 2 identical cells, we're going to make 4 haploid cells. Now, if you look, it's really the same amount of genetic material. It's just being partitioned out in a different way. And that's why we need to go through the whole mechanism of mitosis and meiosis and see how they differ. And, right now I want you to focus more on the goal, the rationale behind each process. So be very, very clear. Really, meiosis only occurs in sexual organs. When I say sex organs, I mean those that are producing gametes. In humans, that means testes if you're a male and ovaries if you're a female. I don't know if you often think of plants as having sexual reproduction, but remember those pea plants, the sexual organ of a plant is the flower. So in that case, in a pea plant or a tree or whatever the heck it is, meiosis would only be occurring in the flower, in the specific male and female parts of the flower that we don't need to go into right now. So also remember this. So in humans and other multicellular organisms, mitosis occurs in sort of 2 contexts. One is during growth from a baby to an adult, or if you want to take it to the ultimate place, we all started out as one cell, right? A zygote that just kept dividing and dividing by mitosis over and over and over again. And that really accounted for the increase in size. Probably an adult human has something like 30 trillion cells in your body. They all came from one. So that was all mitosis, mitosis, mitosis. As we just said, an asexual process where identical cells are produced. As an adult human, mitosis still does occur on a limited basis. We're not really growing in height anymore. For example, many of you know that your skin cells, as they get away from the blood supply, die and they fall off, and they're replaced from the bottom by new ones. And that's a mitotic process that goes on during your whole lifetime. The same goes for blood cells, which are constantly being renewed. So in the bone marrow, there are stem cells that are able to undergo mitosis on a continual basis. So I guess you could say that mitosis is important for growth and sort of maintenance in a adult multicellular organism. Okay? One has to do with a sexual life cycle, which is meiosis. One does not have to do with a sexual life cycle. So in the rest of this session, we're going to focus on mitosis, and next time we'll focus on meiosis. But these initial thoughts sort of apply to just getting a grip on the whole thing. Now, mitosis is a part of the cell cycle. It's not really a perfect analogy at all, but you can sort of think of it as like humans and most organisms have a life cycle, right? We're born, we change in certain ways, ultimately we die. Cells also have a sort of cyclic nature to their being and the way they progress through their lives. So mitosis, this is really not the best analogy, but you'll get my idea. During the course of your life, you may elect to reproduce, right? That's one activity you can do. During the rest of the time, you're doing other things like watching genetics videos and things like that. In the same sense, in a similar sense, mitosis is the reproduction of the cell, and that's part of its cell cycle. So sometimes cells are reproducing, sometimes those same cells are doing other activities, right? So the rest of the cell cycle, in other words, the part of the cell's life that is not mitosis, that's referred to as interphase. It highlights the importance of the reproductive process, but really for most of the time, even in a cell that's reproducing over and over and over again, most of the time it's an interphase and it's, not specifically doing reproductive activities. Now, the next part is perhaps a little bit obvious. I say at any time a cell's either dividing or it's not, right? So what that means is either the cell's in mitosis or it's an interphase. And I'll show you a picture in a moment. But interphase itself can be broken down into three major subphases. Those are referred to as G1, S, and G2. So we should be aware of what those mean. G1 phase, or gap 1, some people call it growth. It depends who you ask and what textbook you read. But what's going on in G1 is basically the cell's living. It's doing whatever duties, if you want to think of it that way, that particular type of cell has. If it's a muscle cell, it's contracting and doing its thing. If it's a connective tissue cell, it's, they do various things, but maybe it's making some proteins, putting it out into the matrix. My point is, cells are just living their life, almost like you're living your life right now. I hope you're not reproducing right at this moment. That's what G1 is all about. Cell growth and maintenance is the way it's described here. But when I say cell growth, be careful. I don't mean in a reproductive sense. I mean maybe the actual cell is growing a bit in size. It's doing what it has to do. Glycolysis is going on. It's making energy. It's building proteins. It's doing all that great stuff. Now, we'll see in a minute that G1 is sort of a stable phase. If a cell is going to divide by mitosis, then it goes into S phase. And once it's in S phase, it's committed. And what I mean is, after S, there's no going back. You have to go ahead and go through the cell division process. Now, S phase, the S in the S phase is for synthesis, because DNA is being synthesized. Now, perhaps I shouldn't refer to DNA at this point, but the other way to think of it is that the chromosomes are copied or doubled. So we're going to see a really good picture in a minute, but you may be used to seeing chromosomes in a certain way. I'm not at all a good artist, but before S phase, if you could visualize a chromosome in this way, it would sort of look like that. Not really, but this is the idea. After S phase, that chromosome is doubled and it takes on this appearance of looking like an X, which is the way we're so used to seeing them. What this really means is that pretty much any time you see a chromosome, it's after S phase. The only time we can really visualize them well is when the cell is in the middle of mitosis. Okay? Now, G2 phase only will occur after S phase in a cell that's about to divide. So during G2 or GAP2, again, the cell is doing its thing. It's, growth and maintenance just like G1. There are also some specific things that occur for the cell to get ready to divide. Organelles divide, special proteins needed for the actual division process get made, etc. Okay, so let's take a look at a picture, because they say a picture's worth 1,000 words, right? And I really like the way this one came out. So up here, M phase is basically mitosis. It again depends, unfortunately, on the textbook or whoever you speak to, but the way I look at it, M phase contains not only mitosis but cytokinesis as well. Don't worry about that right now. Just think of that as mitosis, okay? And since it's a cell cycle, a cell that's dividing constantly is going to be going around and around and around this cycle through G1, through S, through G2, right? Interestingly, though, a lot of cells in your body have decided, if you will, not to divide anymore. More accurately, they've been told not to divide anymore. Cells in your nervous system, for example. So think of this. G1 is a relatively stable state, and sometimes cells decide pretty much that they're going to stay in G1 for the rest of their life. If that's the case, we say that they leave the cell cycle, and you see that little arrow there. I've drawn over it a little bit, but it says G0. So when a cell really basically decides it's not going to reproduce anymore, it's just going to live its life and do its thing, it goes into this state called G0, which really is G1 extended indefinitely. So if the cell is in G1, if it's said to be in G1 as opposed to G0, there's sort of an implication that it's a cell that is constantly dividing, perhaps some type of stem cell that's going to go through G1, S, and G2 and then do it all over again. Right? So also the reason I like this diagram is because it shows you very clearly that interphase, which is drawn in green here, includes G1, S, and G2, and that mitosis and cytokinesis are in the so-called M phase. Right? So this is a very good thing. I mean, memorization is always a part of this terminology, but I need you to understand the flow of what's going on here and the logic of it, right? You need to go, in particular, you need to go through this S phase if you are going to reproduce. If you still don't get why, it's okay. We're going to go through that in more detail in just a moment. Now, as we look to go through the mechanics of mitosis, mitosis traditionally consists of 4 phases. And I say or 5, sometimes 6, 7, 8. It really depends how detailed people want to get into it. You need to know the 4 phases. I'm adding one extra just because I think it's an instructive part of it. Guys, really what you need to realize, though, is this mitosis is a process, right? It's unfolding. It's not stop, start, do this, do that. So we've sort of named the phases according to, different things that are going on and that we see during this whole procedure. It's not really like there's a very defined start and stop to every one of these phases. So also importantly, it's a nuclear process. Now, nuclear means pertaining to the nucleus. This may seem overly technical, but the chromosomes in a eukaryotic cell are in the nucleus, right? So that's all these events that we're talking about occur in that context, in the nucleus. If it's not going on in the nucleus, it's really not technically part of mitosis. And we'll see in particular something towards the end where you can see why this is relevant to our discussion. So it's a good idea, not only is it a good idea, I'd say it's a necessity to memorize the four major phases, which are prophase, metaphase, anaphase, and telophase. Prometaphase I put there in parentheses because we're going to take a look at it, but it's really going to depend on what your textbook/instructor wants you to know. And I just want to emphasize, we're going to go into mitosis now, but remember, the purpose of it is either growth, and when in that sense I mean growth, say from a baby to an adult, or leading to asexual reproduction in protozoa or the like. All right? Well, let's get to it. Before we really can, I want you to take a look at this beautiful picture of a chromosome. Now, when mitosis is ready to begin, remember, if you're about to go into M phase, you must have already gone through S phase. So the chromosomes have been doubled or duplicated. It's very important to both understand why and what this means in terms of the structure of the chromosome. What it means, number one, is, remember when I said if you could see a chromosome during G1, it would look basically like what this blue box contains, okay? That's one full chromosome when it doesn't have to worry about dividing or replicating or anything like that. It's also called a chromatid, so this terminology gets a little annoying to some, but when a cell's not dividing, chromosomes consist of one chromatid. However, after S phase, basically what happens is an exact copy of that chromatid gets made. And here it is. So this is what gives this X-looking appearance here. And these are called sister chromatids because they are literally identical. We've copied it. And the bottom line, if you understand this, guys, you'll understand mitosis, at least in its essence. In order to make, remember that little picture we drew where we said there's one parent cell and we're going to make two identical daughter cells? Well, the parent cell would not have enough genetic material to partition out to those two daughter cells if it didn't duplicate it. That's the whole point of this. So we're taking and we're making it double, and then we're going to pull these apart so that each of the new cells gets enough, gets a full complement of that information. Terminology-wise, for better or for worse though, once this is doubled, it's still referred to as one chromosome. Now it's simply a chromosome that is composed of two chromatids, sister chromatids. Okay? And also you might want to take note of this relatively central portion. In reality, it can be located more toward the top or more toward the bottom. A lot of them are central, called the centromere. We'll have more to say about this in a minute, but this is the region where the two sister chromatids are held together. It's also basically the region where spindle fibers are going to attach to this chromosome during mitosis. Now, chromosomes are not, I kept saying if you could see this chromosome during G1, they're not visible during interphase, and I guess I should have been more specific, particularly during G1 of interphase, but even during S phase and G2, they haven't really fully condensed into this. I think of these type of condensed chromosomes almost like a spool that the DNA is wrapped all around. So we can't see them until they become fully this way. When they're all diffuse and they're just, not really individually visible under the microscope, they're referred to as chromatin. So that's why I say whenever you see these beautiful pictures of chromosomes, it's almost always during mitosis, because that's the only time they gather themselves up into this beautiful X-like structure. So if we try and, I think we will succeed as well, connect the cell cycle to what's going on with the chromosomes. First of all, this is important. So we've made a hypothetical organism here. This is one homologous pair that we've drawn in blue. This is a different homologous pair that we've drawn in red. And we only put the A's and the B's there to show that, gene A is on one chromosome. It must be a different one than the other one. So look here, in G1, if you were to look at these chromosomes, By the way, this is a hypothetical organism where 2n equals 4. In other words, 1, 2, 3, 4, you can see them, and n equals 2. This is usually what's done because you can imagine if we put all 46 human chromosomes there, it would get very overwhelming to try and think about them or keep track of them. So this is a hypothetical organism, but just think of it as a great example so you can understand what happens no matter how many chromosomes there are. The bottom line of this picture is that after the S phase, if you were to see them when they're ready in G2, you would see that each of them has been doubled, right? Now 2n still equals 4 and n still equals 2. The difference between these chromosomes pre-S phase and post-S phase is simply that they have been duplicated so that there's now enough genetic information to partition out to the 2 new cells that are going to be created during mitosis. So the next thing we want to do is take a little bit of a detailed look at mitosis and the processes that occur during it. One thing I have to say, which is not ideal, but it's the way it turned out here. Here we made 2 members of a homologous pair the same color. In the diagrams that follow, it's going to be set up a little differently. In fact, so I don't want you to get confused between the two. Let's take a look at this diagram and get our bearings. This is still a hypothetical organism, a hypothetical cell, where 2n equals 4. But what we did here, I think, is a little better in the sense that the 2 large chromosomes, these are one homologous pair. One's blue and one's red. So what the colors mean in this context is that the red, for example, came from the mother and the blue came from the father. So just like we were saying at the beginning here, you could think of this in 2 ways. The way I've drawn it here, you can think of this as 2 homologous pairs for a total of 4, right? I'll use red ink. Another way you could look at it is everything in blue is one complete set, which happens to only be 2 because this is that hypothetical organism. And these are one complete set, right? You can tell who are homologs because they're the same size. You can tell by the color who came from which parent, which chromosome, all right? So you have to realize now we're beginning mitosis, so we're in prophase, and the chromosomes have already been doubled, right? We've gone through the whole interphase, including S, and a couple of key features happen here. So even though we're drawing them already condensed, the point is the chromosomes are fully condensed at this point. You can see them under the microscope with that X-like appearance. Other organelles that you may or may not have heard of before called centrosomes are beginning to migrate. Here's a centrosome, and you can see that what a centrosome really is a pair of organelles called centrioles. I don't want to get too detailed about this, but what a centriole basically is a microtubule organizing center. If that doesn't mean anything to you, don't worry about it. It means it's a place, though, that can make microtubules. Microtubules have many, many functions in cells, but the spindle fibers that are going to reach out and touch the chromosomes are microtubules as well. So it says that the centrosomes are migrating. They started off in one spot and they've already migrated apart from one another. Ultimately, we're going to find that they're going to be on 2 sides of the poles of this cell. A lot of times the analogy, thinking of this cell as like the planet Earth, you'll have your North Pole and your South Pole, and the middle is called the equator. So these spindle fibers, so-called, you're starting to see already coming out from the centrosomes. Soon you're going to see them contacting the chromosomes. Okay, one other thing that we should mention since it's on this diagram, and this is also something that's going to depend on the way you're taught it. The term kinetochore and the term centromere sometimes are used interchangeably. Technically, this is not correct, but it really depends how deeply you want to go into it. So I'll just tell you the truth, the technical truth. The centromere is the central portion of the chromosome, but it's made of DNA. It does hold the 2 sister chromatids together. The kinetochore is a protein region that's associated with the centromere, and it's really at the kinetochore where the spindle fibers attach. So the 2 structures are intimately connected, one DNA, one protein. They're both right at the same region, and that region is often considered as a sort of single entity, just in case you need to know it that way. But if you just hear the term centromere, there's a lot of centras here. Centromere, and no one ever says kinetochore, then don't worry about it. If they say it, you want to be able to know the difference. All right, so it's sort of a mess, and what I mean by that is there's no organization or sort of order to the way the chromosomes are lining up here, but that's what we're going to start to see, and this is why I wanted to show you pro-metaphase. So if you don't have to know about this particular stage, that's fine, but what's important to note is what's going on here. So it actually started doing it in prophase, but the nuclear envelope you can see is gone here. All right, it's broken down. The reason is these chromosomes are on the move, and eventually when we're done, we're going to have two new nuclei that we make, but we have to get rid of the original one in between so that the chromosomes can move. Very important. Spindle fiber formation occurs. You guys can see that the centrioles or centrosomes have fully migrated to what would be the equivalent of the North and South Pole here, and they're sending out spindle fibers. What you need to get out of this is that from the South Pole, if you want to think of it that way, you'll see if you look out, there is a spindle fiber connecting to every single one of those 4 chromosomes. And the same goes for this pole. There's a spindle fiber connecting to every one of the chromosomes. So they're being grabbed from both ends. And this is very important. This is happening between prophase and metaphase, thus pro-metaphase, right? Now think of what happens if now, I'm not much of a fisherman, but say you were fishing and you cast out your reel. That's sort of like the spindle fiber going out. And you hooked onto a chromosome. But someone else on the other side had hooked onto it as well. If you both reeled it in, you can imagine what would be the result. Well, all the chromosomes, assuming the forces were all equal, would line up right at the mid part of the cell, the equator, and that's exactly the definition of metaphase. But as I said, it's a process, but here's the hallmark of metaphase. You see all 4 of the chromosomes are lined up right at the so-called metaphase plate, or it's very appropriate to also call this the cell's equator if you're using this sort of planetary analogy. So that's what metaphase is. You can see clearly under the microscope when a cell is in metaphase, and I'm going to draw something on here now. If you understand this, you understand all of mitosis, I believe. If I can get a line right in between the middle there, if we want to make 2 new cells, all we got to do, guys, is grab one sister chromatid and bring it toward the top, grab another one and bring it toward the bottom, and do that for, in this case, all 4 of the chromosomes. But however many they are, that's what we're going to do. Can you see that if we do that, we'll have 2 new entities, each of which they're identical to each other. This is going to come out in the next picture even more. But that's basically it. We got these lined up, and we're going to split them and divvy out the material to 2 new cells. In anaphase, the hallmark of that is that the sister chromatids separate, and the reason they're separating is there is actually enzymatic activity that dissolves the connection of the sister chromatids. And to continue with the fishing analogy, now if you start reeling them in, they're not attached to each other anymore. Half of them are going to go towards one pole, and half of them are going to go towards the other. Now I know you'll be able to see. All we have to do at this point is just cut that right there. And can you see that we'll have 2 cells that are genetically identical? Look, I mean, however way you want to count it. There are 2 red ones on top, 2 red ones on the bottom. There are 2 big ones on top, 2 big ones on the bottom. This is it. So we just have to finish up the process. This is anaphase. And the last one is telophase or telophase. Tele just means the end, sort of. So what we have here, you can definitely see 2 new cells in the beginning stages of their formation. We've got a reformed nuclear membranes, which are here, around the new set of chromosomes. You're not going to see it on this picture, but ultimately, if this is going to be done, the chromosomes will decondense back into chromatin. The centrosomes are no longer needed, so they go away. And basically, again, please check with me. This cell is genetically identical with this one, right? If you have to pause it and just look, convince yourself, they're genetically identical to each other. And furthermore, it's worth asking ourselves, because I said it, these should be genetically identical to the original parent as well. Well, are they? You may be thinking not so much, because let's go back and look at the original parent. Here it is, even though I wrote all over it. It doesn't look identical, but what's the only difference? The only difference is, look, here there's two big ones and two small ones, two red ones and two blue ones, but they're duplicated, right? Here, they're not duplicated anymore, and there's a good reason for that. When would be the only time they would be duplicated? If they're going to go through the cycle again. So if we think of the cell on the top, if in fact it was going to go through S phase, that's when you would see it getting duplicated in that fashion, and these ones getting duplicated in this fashion, and then it would literally look like the original, right? The original parent. But that's only a matter, remember, the doubling only occurs as a prerequisite to a further division. So it's very important to understand that. If you think of the problem, if you look at the problem-solving ones, if you don't get this right now, it's okay. But at a certain point in time, if we go back to this original parent, we'd have to say there's 4 chromosomes in that cell, right? But there are 8 total chromatids, and the reason is those 8 chromatids have to be partitioned up into these 2 new cells. Let me erase my beautiful artwork. This is the same amount of genetic material as was in the parent. There are 8 chromatids, but now they've been separated correctly. And if these cells were to divide again, then you'd see those X-looking types of things. Now there's one more thing that has to happen here, and it's pretty simple, which is that you see a cleavage furrow is forming there. If you just separate these 2 cells apart, now they're 2 completely independent individual cells. But here's the reason I mentioned before. The process that causes them to split like this, which you can see here, is called cytokinesis. That literally means moving of cells or something like that, but it's the act of cutting it. Cleavage means cutting, right? It's the last event in cell division, and it is necessary for mitosis. I mean, it depends on the exact type of cell we're talking about. In plants, for example, the cells end up physically staying together, but don't worry about that. Cytokinesis is not considered to be a part of mitosis. Why? Because remember that technicality we said, that mitosis only pertains to nuclear events, events having to do with the chromosomes. This is important, but it doesn't have to do with the chromosomes, and it's taking place, the cutting, outside of the nucleus, right? The cytoplasm is being cut. So, that's a technicality, but it's an important one sometimes on an exam to understand that cytokinesis is technically not one of the phases of mitosis. Even though, and not everybody does it this way, we included cytokinesis along with mitosis in the M phase. I think it's rational to do that because the M phase, the whole point of it is to achieve cell division, and you need cytokinesis to achieve that. Just be careful and realize that technically it's not part of mitosis and it's not one of the phases of mitosis. Well, that ends our session about mitosis. We need to address meiosis next, and I hope you'll join me for doing that.